thank you here on main campus for coming today. Today we have one of our new faculties again in Barshop, in the Barshop Institute, Lizhen Chen. She's recruited here and started in May 2016, right? Lizhen got her Ph.D. from the University of Georgia, then moved on to UC San Diego to do her postdoc, from where she is now started, I think, a lot of the projects that she's working on now in the microtubule, regulation of neurons and aging now, I think, using the worm system. So she's actually, I was kind of impressed by the number of first author papers on her CV rather than a lot of the middle author papers and things. So almost all of them were first author papers through this career so far. So we got a little flavor at the TRP seminar a few weeks ago from her postdoc that she's training on what she's working on, but I think hopefully this will be a little expansion of both new and old. And thank you again, Lizhen, for agreeing to talk, and looking forward to it.
I don't think this is working. So.
Well, maybe it's because the offsite is not working.
So thanks, Adam, for the introduction, and thank you very much for coming today. So before I start, I want to briefly mention this background image. This is actually generated by a summer student in my postdoc mentors lab, Alex Tazuva. She penned this for me when my first paper was accepted, so I would really appreciate it. I think it nicely illustrates my research interest, which is to understand the toolbox that neuron use to maintain its function, particularly in response to aging and injury. I'm not sure if we should.
Yes.
So as stated in the title, so I will be talking about two different tools from that toolbox. One is microtubule regulation, and the other is alternative splicing regulated by self-RNA binding protein in axon regeneration, and more recent extension of the study to neuronal aging. So first, why do we care about axon regeneration? Because it is the essential step to restore neuronal function after spinal cord injury and other conditions involving disconnection of axons, like stroke. So the patient of spinal cord injury usually suffer from lifelong paralysis, because in our central nervous system, axons do not normally regenerate. So there are 0.3 million people suffering from SCI, but no effective treatment is available currently. But in contrast to the central nervous system, some other neurons, they do regenerate their axons after injury. For example, invertebrate CNS neurons, or embryonic central nervous system in mammals, or even adult peripheral nervous system, they could regenerate the axon after injury, although this capacity dramatically declines with aging. So, what controls axon regeneration? Back in 1980s, some pioneer people have done such an experiment. They graft PNS nerves into injured the CNS lesion site. These mature CNS neurons, which do not normally regenerate, now with this PNS graft, they now can regenerate their axons into the peripheral nerves. So this experiment really suggests that this peripheral graft is providing a permissive microenvironment for these central nervous system neurons to regenerate, and also indicate that the failure of CNS regeneration could be due to the inhibitory microenvironment. And since then, people do identify many extrinsic inhibitors, including no-go, MAG myelin-associated glycoprotein, OMGP. These are membrane proteins produced by oligodendrocyte. Although most of these, they do show that these factors inhibit axon regeneration, although most of them are done in vitro cell culture system. And more recently, when people simultaneously remove all the major type of inhibitors using mouse knockout technique, we expect to see dramatic enhancement in axon regeneration. However, we only see very limited regeneration. Now people start to realize more regulators, especially the intrinsic regulators, remain to be identified. So we decide to do a genetic screen using C. elegans, this powerful genetic mutant, to screen for factors involving in axon regeneration. So this is a candidate gene screen because the whole genome screen wouldn't be feasible. We first chose conserved genes which has been implicated in the nervous system. We obtained the mutants from the gene bank. This is one advantage of using C. elegans because you can get a genetic mutant strand for almost every single gene in the genome. Whatever you want to study, you can always buy a mutant for $7 and then you can study the mutants. So after getting the mutant, we crossed them into our GFP marker so that now these neurons light up. You can see the neurons and the process. And then we use laser to cut this axon. This is a blow-up image showing this laser injury site. After that, we analyze the regenerated axon from this injured axon and compare the mutants to wild type to see whether they have normal regrowth, reduced, or enhanced regeneration. So from this selected pool of candidate genes, we actually identify 70 of mutants who showing significantly reduced axon regeneration, meaning these genes are required for regrowth. At the meantime, we found a smaller number of inhibitors whose mutants show enhanced regeneration. And these include both previously known regulators and novel regulators. For those mutants show a defect, we then examine the regrowth at different time points. So we kind of divide the regeneration process into different phases. First is the injury response phase. And axon will actually respond to the calcium influx and start to retract, and membrane got repaired. And about 4 to 6 hours after that, the regenerated growth horns start to form, and we call this initiation phase and the extension phase following that. So we found that some of the mutants, they seem to be more important for the injury response phase, for example, the MAP kinase pathway, and some of them seem to be more critical at the initiation phase, for example, EFA6, which I will be focusing on today, and prochonin. These are the microtubule regulators, and some RNA binding proteins, and all kind of different functional proteins. So we think this genetic screen is the first time to provide assessment to the broad genetic landscape of axon regeneration. Following that, I moved from screen to mechanistic studies, and I chose the two factors. One is EFA6, which is an inhibitor because loss of function enhanced regrowth, and the other one called ANC75 is an RNA binding protein, which is required for regeneration. So first, EFA6. It's an annotated guanine exchange factor for ARF6. So this protein has conserved the SEX7 domain, which is responsible for the GAF activity. Through a series of deletion or mutation experiment. Well, let me back up. So although I mentioned that EFA6 mutant enhanced regrowth, here is a representative image. It just enhanced the growth length, but the overall pattern remains similar to wild type. And overexpression of EFA6 blocks the regrowth. By doing a series of mutational deletion, we found that the SEX7 domain, either deletion or mutation, does not affect its inhibitory role. Instead, the N-terminus deletion completely abolishes function, and overexpression of N-terminus is sufficient to inhibit regrowth. But this N-terminus really doesn't show a 3D structure or a motif. So this is, we are looking at a graph of disorder probability. So normally a protein with high disorder probability or fragment with high disorder score means they don't have a stable 3D structure and they are more flexible. In contrast, these domains, these all have a very low disorder probability. So this N-terminal, which is the important domain for EFA6 function, seems to be very disordered, except for a conserved 18 amino acids motif. And so this led us to think maybe this flexibility of the N-terminal will allow the protein to interact with different cofactors at different contexts. Then we perform a is2 hybrid screen trying to identify the cofactors interact with EFA6. Remarkably, we identified two microtubule-associated proteins, TACC and DCLK. This is double coating-like kinase. And the interaction between EFA6 and the cofactor is again dependent on the N-terminus. And the conserved motif seem to be very important for the interaction as well. We next look into this cofactor to see whether they also play a role in regeneration. Loss of function show an opposite effect compared to EFA6 loss of function. And these two double mutants resemble single mutants, suggesting that function in the same pathway. And removing DCLK or TACC completely abolish EFA6 phenotype. So these data suggest that TACC and DCLK, which is known to promote microtubule stability, and EFA6 play a negative role here and to inhibit axon regeneration. So now we kind of know EFA6 is involved in microtubule regulation. We wanted to...
Could I ask a question?
Sure.
So in your genetic screen, did the TACC, DCLK come up anywhere?
That's a great question. We did not screen these proteins. The reason is because these proteins are very essential. So these are all temperature-sensitive mutants. And in our previous genetic screen, we picked those superficial wild-type mutants. We don't want to deal with these complicated essential genes. They may interfere with development. So these, we did not even screen them.
Okay.
Sure.
Was that also the case for R6?
R6? Oh, actually, R6 is viable and very superficially wild type. And R6 actually it shows a phenotype similar to EFA6. That's why at beginning we really believe that EFA6 just regulate R6 to regulate axon regeneration, because R6 loss of function also enhance regrowth, but it's not as dramatic as EFA6. So we want to take a closer look at microtubule and see how EFA6 is regulating microtubule. So microtubule is this polarized dynamic structure. They on their plus end, they can grow or shrink. And in some stable microtubule, they have a very slow growth rate, and these some others are showing faster growth. And in adult axons, most of the microtubules are relatively stable with a slow growth from the microtubule plus M. And one microtubule-specific protein called EB, and binding protein, has been used to track microtubule growth. So this protein specifically binds to the growing microtubule plus M. So when microtubule is growing, this protein will bind to it. Once it stops growing or it starts to shrink, this protein will fall off. So EBGFP has been widely used to track microtubule growth. Let me play you a movie and you will see this is a cultured mouse neuron, and you see these comets, and each of these dots represent one growing microtubule plus N. So using the similar technique, we try to image microtubule growth in C. elegans axon, which is much thinner and smaller compared to mammalian neurons, but it works. So you will see a similar kind of dots and these tiny dots. At the beginning, you see a brighter image because with time it got photo bleached. So we then generate chemograph from this kind of movie, so we can see this plus N better. Now in this chemograph, each of the dots now become a line. In EFS6 loss of function mutant, you see significantly more growing microtubule plus N, while in the cofactor TACC and DCLK, they show this opposite defect, and double mutant resembles the single mutant. So this phenotype is consistent with their effect in axon regeneration. And together with some other data that I don't have time to show you, we think EFS6 inhibits axon regeneration by inhibiting axonal microtubule growth. In intact, mature axons, these microtubules are relatively stable with slow growth from the plus N. Immediately after injury, calcium triggers microtubule depolymerization, and a few hours after these, microtubule needs to be reorganized, and microtubules start to show fast growth from their plus N, and this is very important for the initiation of axon regeneration. And we think EFS6 blocks this fast microtubule growth, and we also have evidence supporting that EFS6 may promote microtubule depolymerization at the injury time point. All right, because misregulation of microtubule is often seen in age-related neuron degeneration, and a best example would be the function of tau to stabilize microtubule. When tau is misregulated, microtubule will become destabilized, and this is often an indicator as well as a contributor to age-related neuron degeneration. Since we know EFS6 is important for microtubule regulation, we wonder whether EFS6 could play a role in aging as well. So we examined the age-associated neuronal morphology change. These are wild-type touch sensory neurons at young age, and these are the older age, but still wild-type. So you can see at the younger stages, these neurons look healthy and smooth, with smooth process. With age, many of the axons will become beaded, and sometimes you also see ectopic branches. So we think this can be a marker of neuronal aging. And we found that EFS6 mutant can significantly delay these age-associated morphological changes. Here, same as 12 days, you see more of these defects in wild type, but significantly low occurrence in EFS6 loss of function mutants, suggesting that EFS6 could play a role in neuronal aging. And then I-Ping joined the lab as a postdoc in August last year, so she took over this project. She found that age-related mobility decline can also be rescued in EFS6 loss of function mutants. So she recorded these movies. These are animals in liquid. So you will see they tend animals move much slower compared to young animals. Well, these EFS6 mutants, they are much faster and healthier even at day 10. So this is a phenomenon that also seen in DAF2 mutant. This is a ortholog of insulin receptor in worm, and DAF2 mutants, they are very well known for the long lifespan. So I've been quantified them and found that EFS6 loss of function can partially delay or rescue the mobility decline with age, which is similar to DAF2, but not quite the same. So
Excuse
then,
me.
Sure.
This is something one often sees in interventions and aging where the decline rate itself continues. You've just ramped up the initial function. I even saw that, I think, in your video. The young animals had a higher activity as well, right?
Yes.
So in a sense, it's sort of like osteoporosis in men and women. Men have a higher bone density than women do, and women then get the characteristic deleterious effects of osteoporosis sooner than men. And actually, they have a slightly faster rate of decline, but they both decline.
Yeah, that's interesting, but this is not unusual, right? This happens a lot, but we don't really see a exact correlation or between this younger age mobility and lifespan. So, for example, some mutants, they could be hyperactive at young stage, but they don't live longer, right? So, that's true. Okay, just because the similarity between DAF2 and EFS6 in this mobility, the phenotype, I've been wondering whether EFS6 could also modulate lifespan.
So in your video here,
Oh, sorry.
the mutants of EFA6, if they move faster, is this centrally controlled from the nervous system, or it is a spontaneous musculature function?
Okay, maybe in 2 slides I will show you.
Oh, I see.
Okay, so I've been tested whether EFS6 loss of function could live longer, and indeed she found that this mutant can significantly live longer, especially for the median lifespan, if not the maximum lifespan. And she also found that this age-associated decline in reproduction seemed to be partially rescued in EFS6 mutant. So when she was doing this lifespan experiment, she has to move the worm to fresh place every other day. So she found that at later stages, the EFS6 plates, they usually have more eggs compared to wild type. Then she started to quantify them and found indeed at older stages, EFS6 mutant shows significantly more progeny, although the overall total progeny number doesn't show significant difference. So this indicates that EFS6 might also involved in regulating reproduction and health span. Oh, now she asked in what tissues EFS6 plays its function, just to answer your question. So she compared the transgene expressing EFS6 in neurons versus expressing EFS6 in muscles. She found that only the neuronal expression of EFS6, which is this red chase, can completely abolish EFS6 loss of function mutant phenotype in lifespan, while overexpressing EFS6 in their muscle tissues cannot do so, suggesting that these protein might function in the nervous system to regulate the lifespan. And more complicated to interpret these mobility data because she found that neuronal expression can rescue mobility, so does muscle expression at early stages, but not at the later stages. So maybe EFS6 also play a negative role in muscles, in this case, in these trans genes. All right, so microtubule mutants get involved in lifespan regeneration. EFA6 is not the first one. In 2013, there was a paper from Nicholas' group from Australia. They found that PTL1 mutant, PTL1 is the ortholog of tau protein in worm. They found that loss of PTL1 significantly shortened lifespan. So I've been wanting to see whether EFA6 could, and PTL1 show similar well, we know they showed opposing effect. She wanted to see whether PTL1 is required for EFA6 function, and she generated this double mutant. And from this genetic interaction experiment, she found that PTL1 EFA6 double mutant seem to lay in between PTL1 single and EFA6 single mutant. So this is actually suggests that EFA6 and PTL1, they play opposing role in regulating microtubule stability, and this is also reflecting their function in regulating longevity. So in addition to PTL1, IPIN also checked another microtubule regulator called protronin. So protronin is a microtubule -N binding protein, and it binds to -N to stabilize microtubule. Loss of protronin, similar to PTL1, also shorten lifespan, although it's not quite dramatically. And the EFA6 protronin double mutant lay in between again. So again, this support the model that EFA6 inhibit microtubule stability while protronin promotes microtubule stability, and this somehow can regulate longevity. So next, IPIN took a pharmaceutical approach in addition to this genetic manipulation of microtubule. So she treated the animals with microtubule drugs. First, she treated with the epsilon B, which is known as a microtubule stabilizing drug. She found that this stabilizing drug can prolong the lifespan, although she is repeating this experiment again because this curve doesn't really look very typical to us. But at least this preliminary data support that if you stabilize microtubule, it could extend lifespan. She also used a microtubule destabilization drug, nacardazole, and she found that nacardazole can completely abolish EFA6 effect in lifespan regulation. Again, supporting that EFA6 regulate lifespan through regulating microtubule stability. And following that, IPIN wants to understand whether this microtubule regulation and how does it interact with other aging pathways. So these three transcription factors function downstream to the DAF2 signal pathway, and these are required for longevity regulation. Loss of function of these mutants significantly shorten lifespan. So I've tested the double mutants of EFA6 and these transcription factor mutants, and found that the heat shock factor 1 mutant can completely abolish EFA6 effect, and now the double mutant resembles the heat shock factor single mutant, suggesting this effect is dependent on heat shock factor 1, while DAF16 or SKIN1, this double mutant lay in between the single mutant, suggesting more a parallel pathway. So together, our preliminary data seem to suggest that stabilizing neuronal microtubules can delay neuronal aging and promote longevity in a His-Shock factor-dependent manner, and microtubule regulation could play a role in this His-Shock response, His-Shock stress response. But exactly how microtubule regulate neuronal function and then regulate aging and and how these factors interregulate each other remain largely unknown. So this will be one of our major focus in the lab in the coming few years. All right. So now I'm going to. Hi, Carl.
So you think the dependency on HSF1 is because EFA6 is chaperone-like, or you think it's downstream products of HSF1 transcription that help stabilize?
At this moment, we have no idea exactly what's happening, but there is something interesting I probably have mentioned to you earlier. So the EFA6 mutants, they are heat sensitive. Although at 20 degrees, they seem to show this longer lifespan, but we have tried acute heat shock experiment to heat shock them at 35 degrees. They actually die faster than wild type. So yeah, something might be going on, but we don't know yet at this moment. All right, so my second part will be focusing on alternative splicing regulated by self-RNA binding protein. Alternative splicing is a fundamental mechanism regulating gene expression and generate molecular diversity. So one single gene locus is able to produce functionally distinct proteins through alternative splicing. And in our genome, more than 90% of multi-exon genes are actually alternatively spliced. So, and this alternative splicing is regulated by specified RNA binding proteins. And the one I'm going to focus on is called self, CUG, BP, and LV-like family RNA binding protein. They have 3 RRM domains. These are the RNA recognition motif. And again, very complex in mammals, 6 orthologs, while in worm there is just 1, and in fly there's only 1, making it much easier to study them in these model organisms. So this is the image of worm self-RNA binding protein fused with GFP. They localize in these subnuclear speckles, which is consistent with their, this is where alternative splicing occurs. And they are expressed in the nervous system, same as the mammalian self genes. And this mammalian self gene has been linked to neuronal disorders like seizure, schizophrenia, and Alzheimer's disease. Like I mentioned earlier, this is also identified from our previous genetic screen. So we actually screened a few, well, probably 20 or so RNA binding proteins, and we found these are required for exon regeneration, with self mutant showing the most dramatic defect in regrowth. That's why I was very single-minded. I just chose this protein for further study. And this is a representative image. These exons, they seem to be able to form this regenerated growth horn. However, they cannot extend. In order to understand how self is involved in exon regeneration, we perform RNA sequencing, trying to identify genes with changed expression level in this mutant. And we also perform clip sequencing to identify mRNAs directly bound to this protein. And we actually deduce these consensus, which means in worms, UNC75 or self protein prefer to bind to this GU-rich motifs. We then compare these genomic approaches with our previous regeneration screen to see what are the functional targets of self, really involved in axon regeneration. And we found one group of genes really stands out, which is synaptic transmission genes. These genes are required for axon regeneration, and they show up in both of our RNA sequencing target list and our clip sequencing target list. UNC64 syntaxin is one of the targets identified. So syntaxin mediates the interaction between synaptic vesicle the fusion and the exocytosis of the synaptic transmitters. And this gene is alternatively spliced. They have this last axon encoding the little transmembrane domain. They are alternatively spliced, and all the rest of the gene is identical. And UNC64 syntaxin is required for axon regeneration. So loss of function should be decreased axon regrowth, although this is not as dramatic as the self-mutants. To understand how self-regulate syntaxin regulation expression, I generate these reporter gene to fuse the RFP to the A isoform and GFP to the B isoform so that we can study the alternative splicing of UNC64 syntaxin gene. So these are the wild-type animals. You can see the majority of UNC64 expression is from the A isoform. You barely see any GFP expressed here. When we take away UNC75, which is when we cross this reporter to UNC75, the self-mutants, we see a very dramatic difference. Now you see a decrease in A isoform expression and an increase in B isoform expression, supporting that the normal function of self-RNA binding protein is to promote A isoform and inhibit B isoform expression. And our clip sequencing also mapped the binding site of self on UNC64 gene. So we've mapped it into the intron between the A and B axon. And I generate another reporter gene. Now this GFP is only report for B isoform expression, and A isoform we don't know what's going on here on this reporter. So again, consistently we found when we lose UNC75, the RNA binding protein, B isoform expression is increased, suggesting that UNC75 inhibit B isoform expression. And when I delete this binding site here, same reporter gene, when I delete the binding site, now we see a similar effect as UNC75 mutant, even in wild-type background. So either you remove the protein or you remove the binding site, you see the same effect. So this really tells us that UNC75 regulate UNC64, the syntaxin, and the syntaxin could be a functional target mediating self-function in axon regeneration. So to test that, we overexpress syntaxin in the self-mutant background. So loss of UNC75, the self-RNA binding protein, you have this much of the axon regeneration. Now, if you overexpress syntaxin in the background of UNC75 mutant, you can significantly, although not completely, rescue this defect. But you don't see the same effect with UNC64B isoform or protein without the transmembrane domain. So this really confirm that syntaxin might be one of the self-targets mediating its function in axon regeneration. Now we decide to look whether its function is evolutionarily conserved. A quick, easy experiment is to express a mammalian self-protein in worm to see whether this mammalian protein is able to rescue the worm defect, a worm mutants. And I picked 2 out of 6 because these 2 are closer to each other and these 4 are in another subgroup. So I picked for self-2 and self-4 and expressed them in the worm mutant. And indeed, overexpressing or expression of the mammalian self-protein is able to significantly rescue but not completely rescue. It could be due to codon usage and expression issues, but at least this tells us the function might be conserved across species. We then took a step further to look into self-2 knockout mouse. So our collaborator, they generate self-2 conditional knockout allele. Originally, they want to study its function in heart development, but they don't see an effect. But I wanted to look into the function in nervous system. If we delete self-2 in whole body, these animals will not survive up to a newborn. So they die at newborn. So we have to use a tissue-specific Cre to knock out self-2. And first we use nesting Cre, which is considered as a neuronal Cre. And we found even with neuronal-specific deletion, these animals are not very healthy, and they usually die at probably 20 days after birth. But these at least allow me to get some primary neuronal cultures from these animals. And I found that in the self-2 mutant DRG neurons, the axons actually grow much less compared to control. So this really encouraged me to look into the in vivo function of self-2 in axon regeneration. So here I used another Cre, a provalbumin Cre, to delete self-2 in large diameter DRG neurons. And then examine DRG axon regeneration after this crash injury. So compared to the control, self-2 knockout shows significantly reduced axon regeneration, which is indicated here as SCG10 expression. This is a marker of regenerated axon. So again, these provide evidence that RNA binding protein function in axon regeneration is conserved across species. So not only the protein function, we found the regulation regulation on syntaxing gene expression by self seem to be conserved as well. So this is looking at eclipse sequencing data of mouse self-2. And we found mouse self-2 also bind to syntaxing gene mRNA. And this syntaxing 2, very similar to worm syntaxing gene on64. It has 2 alternatively spliced axons at the 3' end of the gene. And self-2 binds to the intron very close to the alternatively spliced axons. And here is QRT-PCR data showing that in self-2 mutant, the isoform 2 expression is significantly reduced. Sorry, this is isoform 2 and this is isoform 1. So again, this suggests that not only the protein function, but also this self-syntaxing regulation is conserved across species. Now to summarize, I want to borrow this graph from a review paper, a preview paper for our research article. So basically we found that an RNA binding protein called SELF or ANG75. It regulates alternative splicing of a set of synaptic transmission genes. And these synaptic transmission genes have different alternatively spliced isoforms and play different role in axon regeneration. In this case, syntaxin A promote regrowth, while syntaxin B doesn't seem to be involved. And one hypothesis is that these synaptic transmission genes could play a role in membrane trafficking or membrane addition, because to repair an injured neuron, you need more, you need membranes to add to the injury site to seal the injury site, and also you need new membranes when the axon grows further. And together with the phenotype in self-mutant, you see they can form regenerated growth hormone, but they fail to elongate further. So we think maybe because there is no enough membrane trafficking to provide enough membrane to support axon regeneration. All right, I think these are my old data studying the SELF2 function in regeneration. And recent studies, G1 studies, really link SELF2 to Alzheimer's disease. So I decided to extend my research into look into the RNA binding protein in Alzheimer's disease. So in the last few slides, I want to share with you some of my research plan, and we don't have any data at this moment yet. So this GWAS analysis nominates CUGBP2. CUGBP2 is another name of SELF2. Because actually SELF2 has many different names, but these are the same thing, identical. So they found CUGBP2 is very significantly associated with ApoE. And this association is actually more significant compared to BIN1 and clustering. And these two are recent risk factors identified for AD. Another independent study also confirmed the association between SELF2 and Alzheimer's disease. So I think maybe SELF2 could play a role. And remarkably, well, probably not very surprising to you as well, we found all those major Alzheimer's disease genes, they are alternative spliced, which is not surprising because 90% of genes do, right? But these alternative spliced isoforms seem to be related to AD pathology. For example, APP, which is the amyloid precursor protein, and the exon 7 encode the KPI domain. And this KPI included protein is significantly enhanced in AD human patients, and these will lead to increased A-beta deposition. And another example is tau, exon 10 inclusion, which has been heavily studied. This inclusion is increased in AD because this inclusion will produce a 4R tau, and this is thought to be related to AD pathology. And many other proteins. These are the gamma secretase involved in A-beta processing, and their alternative splicing seem to be altered in AD patients as well, and the BIN1, which is the recent risk factor. So all these important AD proteins, AD genes are alternatively spliced. And remarkably, all these genes seem to be regulated by SELF2. Although I don't have direct evidence yet, but our ECLIPSE sequencing shows binding of SELF2 on all these mRNAs. For example, APP. This is the alternative splice exon 7. You see a big peak of binding, self-2 binding. And we, in addition to whole cell clip sequencing, we also separate the cytoplasmic and nuclear fraction. So you can see the nuclear fraction of self-2 mainly bind to introns, while the cytoplasmic fraction binds to the 3' UTR, suggesting that it also regulate mRNA abundance. But really, I was very excited about the binding of self-2 to almost all the AD genes. Neuro2A are neuroblastoma cell lines, so they are cell culture system. It's not primary neuron or neurons. They're just neuroblastoma cell lines. And APP, which I mentioned that the exon 7 is important for APP dimerization and regulate A-beta processing, and these could be regulated by self-2. And I want to show you another example, the tau protein and tau alternative splicing. This exon 10, there is the binding of self-2 on close to this alternative splice exon. And there are 6 different isoforms of tau, maybe even more. And people found these 4R tau, including the exon 10. These are the pathological isoform. And previous study have found that overexpressing self-2 in HeLa cells actually can inhibit this 4R tau formation. So this suggests that maybe self-2 could play an inhibitory role in the 4R tau formation and involved in AD progression. So this really lead me to hypothesize that maybe self-2 is a common upstream regulator of many of these AD regulators. When we manipulate a single AD gene, this may not give us a very effective treatment or effective many dramatic effect on intervention of neuronal aging or degeneration. But if we manipulate this common upstream regulator, so now if we manipulate the self-2, now we can simultaneously change the expression of or alternative splicing of all these AD-related genes. Maybe this could give us a better way or better therapeutic target to intervene the neuron degeneration like AD. So this is purely just hypothesis yet at this moment. All right, with that, I want to thank Aiping, who is my postdoc, and also Christina, who is the first member in my new tiny lab and makes my lab lively. And my mentors at UCSD and Dr. Lu's lab here, who is a long-term collaborator, and Dr. Fisher lab for their help with our aging experiment, because we are totally new into this field. And thank you very much for your attention.